There are a number of, again, free licenses, especially if you are from NAIPI. So how do you know if you're a part of NAIPI? There's a website, naipi.org, where you can see the map, and all of the 23 idea states are listed on there. If you're from one of the academic institutions in one of those idea states, you're most likely a part of NAIPI. What you need to check is whether that institution is listed, because NAIPI is an organization that brings together PIs funded through the NIH IDEA Institution Development Award. So if you receive this information from your core, you probably have access to those free licenses. Okay, so what I will try to do in the next few minutes is briefly explain how this training is going to be different from what most of you are probably used to when it comes to online coursework like Coursera. What are the specific benefits of taking this training, and how will you be able to use some of the skills or resources that you will gain access to throughout the summer? So first of all, as already mentioned by Jabs in the beginning, we try to assemble training materials, a number of data analysis tools, curated project data sets, so data sets that are ready for analysis, and interactive sessions that some of them are going to be available as recorded sessions, or if you're available during that time, you can join them live. And this is to make bioinformatics more project-based or oriented around case studies. So bioinformatics is a very broad and dynamic field. There's a lot of new things that come up all the time. And so it's very difficult to imagine someone kind of comprehensively understanding bioinformatics in only 3 months. And so that's why, instead of offering to kind of train you and give you everything you need to know bioinformatics, we're saying this is oriented around case studies. How is bioinformatics used in cancer studies, specifically using transcriptomic data? Or how is bioinformatics used in the study of infectious diseases? For example, if we want to understand genomic and transcriptomic data to study the pathogen and to understand the host responses, right? So those are the case studies. Again, those of you that kind of work already on some of those similar projects, this is the opportunity to take your project and just analyze that data as you go along. And that means that we're going to be dealing with a diverse group of individuals, that some are faculty, very independent, driven by maybe research objectives and questions that they would like to get answered, as well as some undergraduate students that might be new to all of this and are just trying to get started. And so that's why we're going to be offering a number of these different resources so that you can explore the ones that you need on your own time, but also have access to lectures and mentors that can actually help you and guide you and help you overcome technical issues that you might face. So what we've done over the past few years is we have collected industry and academia lectures from a number of organizations, including different NAIPI associated organizations, and kind of prepared a basic package. And that basic package includes genomics, transcriptomics, metagenomics, epigenomics, introduction to data science, and kind of collection of different project examples. In addition to that, we understand that a lot of people might come from a biology background, and they might not be prepared for the technical demands of both learning how to program a specific analysis, as well as setting up that analysis to run on a specific infrastructure, like your own laptop, or maybe your core high-performance computer or something like that. And so because of that, this program comes with another important resource called the T-Bioinfo Analytical Platform. Now, the platform offers a semi-guided experience where you have a number of predefined paths that connect independent scripts into a pipeline. And they come with a lot of computational resources. For example, every account will come with 5 terabytes of storage. You can also, at the end, request for that particular pipeline to be submitted into a list of containerized pipelines that you could deploy elsewhere. And most importantly, you get a lot of support. So if you want to really understand how it works, what are the parameters, how do you change them, how do you run it for this type of data or that type of data, this is what the programs are going to be about. It is to provide you with that kind of technical support. For example, how does this achieve learning goals that we're expecting to kind of arrive at after this program? In many cases, you are probably familiar with figures that represent analyses like statistical analysis, maybe principal component analysis, or maybe differential gene expression, or you might be familiar with mapping and visualization of map reads on a reference genome. So the platform will combine those kinds of analyses into an experience where every point in that process will be color-coded and will come with an explanation so that you can understand what you are doing, change parameters if need be, and also remember how did you achieve a specific result, so it makes that analysis reproducible. Now, in the program, we will use both the platform and then we will look at what do some of these individual buttons do? How do they actually take in some data that was already prepared for that stage? What type of data do they produce? What does that data look like? And what does the analysis try to do? How does it work? And so because we have multiple comparisons in each logical step, you will see how by looking at the analogous analyses that are available, what are the differences and how might you choose the right type of analysis for your specific goal. I'm going to pause here because I have a couple of questions I'd like to answer. So question number one from Anelia: if you are not a part of any of the funded programs, how do you register? I will share again links where you can purchase the program independently, or you can go to your PI or to your university and say, I would like to take this training over the summer. And in a lot of cases, they will offer to pay for that or maybe support you partially. And the second question from Ashley was about this recording being available. Yes, this recording will be available after the webinar, and also is going to be on our YouTube channel. So as I mentioned, beyond this logic, it's also important to at least understand how code works and what are some of those steps that you can take to get started with coding in R and Python, two of the most popular languages in scientific computing. So what we will do is we will actually use several of the examples after the big data is processed and we've kind of distilled it to structured data or tables. We will look at what is the context. So how do we ask questions about a particular data? Then what is the logic? How do we actually understand the steps to arrive at a particular result? Then we will try to practice, and then hopefully that will prepare you to be completely independent, both relying on big data analysis tools, which you will find many of those are available today, but also if you would like to take your analysis further and kind of take a look at becoming more independent and being able to write such analyses yourself. So now let me talk briefly about three of the programs. So the first one I'll talk about is biomedical data analysis using R and Python. This is a program designed for those who are interested to learn how to write code. So in many cases, you will have a particular type of data that is already structured. And for that structured data, you either want to perform a statistical analysis, exploratory analysis, maybe you would like to also use machine learning. So that is what this program is going to cover. First of all, for those of you that are not familiar with R and Python at all, there's a way to get started. Right. And so what we've done on the portal is we've taken some of the basic principles like dealing with objects, loading data into an environment where you can analyze data. looking at specific data sets and understanding structures like matrices, data frames, etc. So those are the things that are going to be covered in that introductory course, and that is available completely for free. If you haven't started with coding, we highly recommend it. We'll help you kind of extract meaningful insights out of complex data, visualize and present them. But beyond that, we're also going to go into kind of a high-level overview of what data science is and how is it applied specifically to biomedical projects. And here we will have two tracks. One is BioML in R and BioML in Python. You can choose either one. And you will see how these are used today when analyzing complex data, but also you will be exposed to a number of standard or what we call conventional machine learning tools, methods that are going to be very helpful to deal with noisy data and data that contains a lot of variability. For example, patient data that contains a lot of variability. So here is a brief overview of the topics that we will cover. We'll talk about data wrangling, how to load, transform, and explore a data set. We'll talk about machine learning and statistics. What is the difference? How do they kind of compare to each other, and why high throughput data presents some additional challenges for these methods? What is the general kind of distinction between unsupervised and supervised types of analysis? How can we use machine learning for data visualization? Then we'll talk exclusively about clustering and another session about classification. We'll talk a lot about how these methods, not only what types of methods there are, but how are they used in example projects. And we'll try to address some biological questions that you might have. So for example, what is the bigger difference between different tissues, or is it between normal and cancer tissue, right? So we'll kind of use those methods to try to connect it back to the types of questions that you might have. Then we'll talk about feature selection and signature patterns, how we can define a particular signature pattern, for example, a marker, and then how can we understand what that marker actually represents, what feature is it connected to. We'll talk about regression and generalized linear models and deep learning, and how deep learning is used. So all of these are going to be covered in a live session. So kind of like what we're doing today. There will be someone speaking, there will be questions will be answered, there will be some exercises, but then you will also have a number of examples and additional projects that you can practice independently. So what is that going to look like? Here's an example of a course. This is a course on PCA. So how do you actually produce a plot, a 3-dimensional or a 2-dimensional scatter plot to understand variance in your data? You go through, first of all, understanding the problem. Why are you using PCA in this case? Then you will learn the syntax of the code. So how can code be written to actually perform this kind of analysis? Then you will be given that code that you can run, and you will be able to modify outputs. So in one case, you might produce a table. In another one, you might produce a visual output, etc. Then you'll be able to play around with that code and know that you can modify settings. And this is very important because in so many cases, once you know that there's a library that you can use, you just need to modify it to apply it to your data and then make it do what you would like it to do. It is a very liberating experience, and you can start going through a lot of examples that people have posted online and read that code, understanding what it was doing, and know how to modify it to apply it to your problem. And then finally, once you go through several examples like that, you will be given some challenges. And those challenges will come as an interactive notebook, and they will essentially be an assignment. So you will start with some beginning. For example, you've learned the syntax, you've applied it to a particular data. Then the coding challenge will say, okay, write this code. Maybe there's a missing portion, or maybe write it from scratch. or maybe even apply it to a number of different examples that are not trivial. They will need some thinking to overcome some issues that you will have. And again, as a result, you will have an interactive notebook. That interactive notebook will be shared with your mentor, and your mentor will be able to suggest some maybe technical issues, how to overcome them, or maybe how to use other types of methods for whatever challenge you are facing. Now, importantly, we will look at some of the ways that these methods actually work and what needs to be done with those methods to overcome common challenges like missing data, maybe unbalanced classes. We'll talk a lot about evaluating the accuracy and precision, how to trust your model, how to deploy your model after you've trained it. All of those kinds of questions is something that we will focus on and kind of see real world examples. So what are the outcomes of such a program? The outcomes of such a program, first of all, some technical skills and kind of reading papers, looking at the methods and knowing what they mean, but also putting them into a logical scheme so that you can kind of go from experimental design and all the way to interpretation through the lens of the analysis process. How the data is prepared for analysis, how is it explored to eliminate maybe artifacts, batches, and issues with the data, how to then perform actual data analysis, choosing between maybe a model that is statistical or maybe a machine learning model, and then how to produce inference, something that you can interpret either in the context of that particular experiment or something more generalizable, something that you could kind of build upon to understand something about the general population. So again, what we will do is we will use some case studies. And all of these case studies are going to rely on omics data. So we will look at diagnostics and cancer biology, single cell and tissue composition, and we'll also look at microbiome and infection. And you'll be able to choose a number of curated data sets that will be provided to you to kind of play around with and develop a project of your own at the end of this course, at the end of this training program. So again, if you have any questions about this particular program, any of the topics that I just mentioned, please add them to the chat box. I'd be happy to answer any of those questions. And I'll just give a moment here if anyone has any questions. Okay, so again, how do you register for this particular program? The way you register is dependent on which organization you're from. So you could be from 3 organizations: LSU, School of Veterinary Medicine. The second organization is LBRN, Louisiana Biomedical Research Network. And the third organization is the National Association of Idea Principal Investigators. So based on which organization you're from, if you were not present in the beginning of the webinar, we kind of went through all the different options, how to register, but you definitely have to create an account on the Learn portal. And once you do, please email us. And you can email to a number of emails here. So Farhana shared her email, or you can simply reach out to this email right here. We'll be able to direct you on your next steps and even have a call to discuss your specific questions. Okay, so Raquel is asking when the course will start and end. So this is a summer program, and so it's going to start June 15th all the way until August 31st and extended until September 15th. So you will have a full 3 months to work through all of these different things. There's going to be a defined schedule of live sessions, but all the live sessions are going to be recorded, and all of them will be shared on the program page. And I will go over the program page at the end of this presentation.